My name is Sudhir Paul. I'm a professor at the University of Texas in Houston, and I will be speaking to you today about the subject of immune protection against protein misfolding. This is a subject of particular interest in the field of aging, because as we age, proteins tend to change their conformation or their three-dimensional shape. And at this time, it is difficult to treat these diseases that are caused by the misfolding of proteins associated with aging. The best example of a disease that is caused by protein misfolding is Alzheimer's disease. In the United States alone, 5.4 million patients have been diagnosed with Alzheimer's disease. And it's estimated that in the year 2050, 16 million patients would be, the incidence would be 16 million in the year 2050. So this disease is characterized by accumulation of misfolded amyloid beta peptide. So this is a relatively small polypeptide, and then a larger protein called tau. And these proteins form plaques, and that's the amyloid, and tau forms tangles in the brain, resulting in essentially memory loss and cognitive failure. This is not the only disease that is found to be associated with protein misfolding. About 20% of humans over the age of 80 suffer from senile systemic amyloidosis, and this involves neuropathy in the periphery as opposed to the central nervous system abnormalities caused by the amyloid and the tau polypeptides. SSA, or senile systemic amyloidosis, occurs mostly in the periphery. It affects the heart, kidney, and other tissues. This is caused by a protein called transthyretin. In familial amyloidopathy, these proteins, examples are transthyretin or amyloid beta peptide, they undergo mutations that tend to make the polypeptides aggregate at a faster rate, and this causes early onset Alzheimer's disease or the senile systemic amyloidosis. Other genes can also be involved. In addition to the genes that encode these proteins, certain accessory genes can accelerate deposition of amyloid in the tissues. An example is the gene for the apolipoprotein E4 in Alzheimer's disease. So thus far, 27 distinct proteins have been associated with disease, with amyloidopathy, and over 40 different diseases have been associated with misfolded proteins. So what is protein folding? The word protein in fact means, in the Greek, protein. It means that the protein, the molecule, tends to be flexible and can adopt varying conformations. However, it is true that under normal circumstances, proteins relax into their most stable conformation. That said, proteins can take alternate states. So these are different energy levels at which different conformations, refolding of different proteins can occur. And if the energy barrier between these different states is too high, this is the energy of activation required to convert this conformation into the most stable conformation. But if this energy of activation is too high, this conversion is impossible to make, with the result that proteins get trapped in their unstable state, and often the amyloids, they are the aggregated proteins. They might be in the high energy state, but they are not convertible back to the soluble physiological state of the proteins. So these, in their soluble form, the proteins are called amyloidogenic. So they have the potential to aggregate and form these amyloid plaques that I spoke of. So there are different kinds of folding motifs. A protein, individual regions within the protein can be disordered, which essentially means it's flexible and it's dynamic, it's moving around, or the individual regions can form alpha helices characterized by this helical structure. The amyloidogenic proteins generally have these beta strand motifs in which different parts of the protein are hydrogen bonded with each other, mostly stretched out, and these are the proteins that from a beta strand they can form this type of hydrogen bonds intermolecularly and form what is called the beta sheets, resulting in aggregation. So there is no physiological function for these amyloids that accumulate as a function of age. The protein folding diseases can affect diverse tissues, ranging, of course, from the brain to the heart, kidney, the gastrointestinal tract, lungs, and essentially the tissue architecture can be destroyed. The anatomic integrity of the tissues is destroyed. There is interference in various homeostatic processes, including blood flow, signal transduction, receptor-mediated signal transduction. The inflammatory status of the organs is changed. There is greater release generally of inflammatory agents, and the amyloid can deposit both within the cells, an example is tau, and outside the cells, and the example there is the amyloid beta peptide. There is no effective therapy or cure, and in one example, the transthyretin amyloidosis, there are statistics available suggesting that about 50% of the deaths that have occurred in the United States of individuals who are 110 years and older has been due to senile systemic amyloidosis. So clearly this, if we want to make people live longer, we would need to find new ways of treating and curing amyloidosis. Now, I'm going to be spending most of my time speaking about Alzheimer's disease, and then I'll offer some comments and some data on another couple of amyloids in addition to the amyloid beta peptide, which forms plaques, and that would be the tau protein in Alzheimer's disease, and then transthyretin towards the end of my talk. So in Alzheimer's disease, the tau forms tangles within the cell. This here is the lipid bilayer separating the intracellular and the extracellular space. This is a neuron within the brain, and an example, a stained neuron is shown in the right upper corner with the amyloid plaque outside the cells and the tangles inside. At any rate, the amyloid beta peptide is this little bitty piece shown in light brown. This is formed by cutting of the precursor protein, amyloid precursor protein, by these enzymes, beta secretase and then gamma secretase, eventually releasing this peptide which forms oligomers. Initially, it's a monomeric state, and this peptide forms oligomers and eventually forms fibrils, and then further large plaques that are visible when a section is made of the brain. So in fact, these are the two hallmarks of Alzheimer's disease. At this point, a diagnostic method for Alzheimer's disease is not available other than cognition tests, but we don't have a clinical non-invasive method of diagnosing Alzheimer's disease. So the definitive diagnosis is done at autopsy, but from cognition tests and behavior, it is generally possible to make a diagnosis of the disease. This is an atomic on the right bottom corner. This is an atomic force micrograph showing the fibrils of the amyloid beta peptide and little oligomers, the little white dots that have been formed from synthetic amyloid beta peptide. So the studies of the formation of these fibrils and these oligomers in the test tube serves as a model for what's going on in the brain. So the peptide, as I said, forms these oligomers that are themselves toxic. These misfolded oligomers formed by amyloid beta peptide aggregation are directly toxic to the neurons. They influence certain tests, again in physiological tests, called long-term potentiation, which is an indicator of memory functions of the neurons. These oligomers dampen down the LTP. And then, of course, the fibrils are the core of the plaques. And these often are thought to be, they're thought to be physically injurious in the brain, although there is some data that they can also be toxic directly for neurons. Now, the misfolding is a function of other compounds in the diluent, in the microenvironment in the brain and also in the test tube. Various compounds like metals can bind the amyloid beta peptide and induce aggregation. Similarly, chemical modification of the amyloid beta peptide with certain oxidation reactions that occur post-translationally can induce amyloid formation. And it takes only a little bit of a misfolded molecule of the amyloid beta peptide to act as a seed around which a lot of additional molecules can misfold. In fact, people make analogies between amyloid beta peptide aggregation and prion disease. The aggregation, in a sense, is transmissible, that if an aggregate is formed, that in itself can induce aggregation of neighboring molecules of the amyloid peptide, and prion aggregation also occurs by a similar mechanism. It is important to remember in terms of immune targeting of these amyloids that these new molecules, the oligomers and fibrils, these are new in the sense that they are not present in youth and they accumulate in old age. So our immune system has not developed tolerance. We develop tolerance to our own self-antigens, but if we are never exposed to amyloid beta peptide in its misfolded state, then we do not have tolerance, so we can make immune responses. And so these are called neoepitopes, new shapes that are formed, three-dimensional shapes of chemical groups within the oligomers and the fibrils that are new, and therefore the immune system can respond. and make antibodies and also cellular immunity against these neoepitopes without damaging the monomeric soluble, the physiological state of the amyloid beta peptide. So indeed, this is what inspired Elan to attempt vaccination against amyloid beta peptide. They immunized humans. This was a phase 2 clinical trial that I'm describing in this slide. And this was preceded by mouse studies, transgenic mouse studies. The mice that were overexpressing human amyloid beta peptide, they developed amyloid plaques in their brains. And when they were vaccinated with the amyloid beta peptide, the mice started making antibodies to the A beta and cleared the A beta. So humans in the phase 2 trial also responded similarly. They made antibodies, and then over several years, studies of the patients in this clinical trial of mild to moderate Alzheimer disease patients who were vaccinated, there was evidence of removal of the amyloid beta peptide. However, the patients, a subset of the patients, about 6–7% of the patients, they showed infiltration of lymphocytes in their brain in what is termed sterile leukoencephalitis. So essentially, there was inflammation in the brain as a consequence of the vaccination. So these trials were abandoned. Now, the company Elan, they proposed that the side effect of inflammation was due to T cell immunity as opposed to the antibodies. So they then said that if we can make monoclonal antibodies and use the monoclonal antibodies for therapy, there is a good chance that the side effects will be avoided. So antibodies are essentially molecules that are capable of specific antigen recognition. They are designed to recognize microbial foreign antigens. However, in this case, as I said earlier, the amyloid beta peptide aggregates have no useful function. So recognizing the amyloid and then removing it is not going to have a bad effect. It's a bad self-antigen, and therefore its removal is likely to have good results. The antibodies are distinguished from other classes of molecules by virtue of very specific recognition of the target, which is achieved by immunizing animals with the target. So amyloid beta peptide was used to induce an antibody to amyloid beta in mice. Then certain sequences of the antibody, these regions in the constant domains of antibodies, the murine regions were replaced with human regions, and a chimeric antibody was produced. The purpose was essentially so that the chimeric antibody would not induce an anti-antibody reaction when used in humans. Again, antibodies, mouse antibodies when injected into humans are foreign entities and can induce an anti-antibody response. And in this case, a humanized antibody directed against amyloid beta peptide, which binds to the variable regions of the antibody shown in purple in this slide. So this antibody was used for passive immunotherapy. A few other comments about antibodies. They are really unique in the sense that there is a very large repertoire of antibodies that all of us make. We can make up to 10 to the power 13 different antibody molecules with different specificity. Now, again, the immune system and antibodies are unique, that they mutate in real time. This happens as an adaptive phenomenon Remember, in general, mutations in proteins are undesirable. In fact, mutations in proteins are often associated with disease, cancer. Amyloid peptide itself, I mentioned that if certain mutations in amyloid peptide can increase the aggregation of the peptide and result in early onset Alzheimer's disease. However, for antibodies, mutations occurring in the variable region, that's good. They are meant to mutate adaptively after they are exposed to the antigen, and the purpose or the function is to develop high affinity binding activity that is specific, in this case for amyloid beta peptide. There are two types of antibodies, what I call traditional antibodies or reversibly binding antibodies. They bind and then they let go of the target. And this trial that I'm speaking of was done with a reversibly binding chimeric antibody. And then later in my talk, I will speak about catalytic antibodies that can work like enzymes. The other drug that is currently under phase 3 trials is Baxter's intravenous immune globulin, also known as IVIG. This is a formulation composed of polyclonal immunoglobulin G from healthy humans. These are humans that are not immunized with anything, and these are simply the constitutive antibodies that humans make that are sold under the name Gamma Guard. This formulation is approved for use, clinical use, for immune deficiency and for certain autoimmune diseases, and it's being tested by Baxter in phase 3 trials for the treatment of Alzheimer's disease. The phase 2 results tended to show an improvement in cognition. In fact, I've seen movies of the trial subjects who became animated after treatment with IVIG, and after a few months of treatment, indeed resumed social activities, involvement in music and more family involvement and so forth. So however, the statistics are not available, and the information at this time is not fully available as to efficacy. No side effects have been disclosed for IVIG. But in general, IVIG is a safe drug. So there is now beginning evidence, there is some initial evidence that antibodies might be useful for treatment of Alzheimer's disease. I think this is worth noting, despite the qualified success that for at least a decade, many trials, clinical trials have been done for treatment of Alzheimer's disease, and unfortunately Alzheimer's disease has proved to be a sort of graveyard of clinical trials. The reason is there is so much going on, so many abnormalities that are going on in the brain of patients with Alzheimer's disease. Not only are there the plaques and the tau tangles that I mentioned, but there's also a heightened state of inflammation. And once the neurons are lost, it is difficult to bring back memories essentially. But the hope is that the immune system can be used to at least retard the loss of cognition in patients and stabilize the patient. So what is the mechanism of antibody action? Antibodies have an Fc component, as I showed you in one of my slides. This is called the constant fragment of antibodies. This region binds to the brain's microglia, so which are the brain's resident macrophages. These immune complexes of antibody with the amyloid peptide, these immune complexes are taken up through the Fc receptors on the macrophages, the microglia, and then the immune complexes are broken up intracellularly, with the result that the immune complexes are cleared, and the result is plaques are reduced in the brain. At the blood-brain barrier, these immune complexes also bind to the Fc receptors, and this time around at the blood-brain barrier, the antibody acts like a pump and essentially transports the amyloid beta peptide from the brain into the periphery. This is shown here. The antibody is injected, a small amount of the antibody gets into the brain. There is ingestion of the immune complexes by the microglia, and the plaques are cleared. The antibody immune complex can also bind to these Fc receptors at the blood-brain barrier, and the peptide is essentially pumped out into the periphery. Yet another mechanism for antibodies that do not get into the brain. Remember, only a very small proportion of antibodies can get in, something like 0.01 to 0.1% of the antibody that's injected actually gets into the brain. So the mechanism, there is for antibodies that don't get into the brain at all, a proposal is that the antibody binding to peripheral blood-borne amyloid peptide, it works like a sink, that the antibody sequesters the peptide in the periphery, and this induces a compensatory release of the brain peptide stores. So as I mentioned earlier, this is really the first indication of some success in the clinical trials with antibodies. There are side effects, one of inflammation, because microglia can release inflammatory mediators upon binding of these immune complexes. So this is a potential side effect. The other side effect of vasogenic edema is thought to be due to deposition of immune complexes in the vascular wall, and essentially small microbleeds occur because of these immune complexes deposited in the vessels, which in turn causes vasogenic edema. So which brings me to catalytic autoimmunity, which in our view can have greater efficacy than reversibly binding antibodies because of the turnover function, and it may be safer also, as I will comment in a few minutes. So I want to give you a background of catalytic antibodies that have come out of a lot of basic science studies over the last decade or so. As in conventional reversibly binding antibodies, these antibodies, which are shown here as antibody AB and then nucleophile nu, these antibodies bind non-covalently and form an immune complex. This is very much like a reversibly binding antibody. However, this nucleophile, a nucleophile is simply a group, a chemical group that likes positive charges. This nucleophile, it can attack this positively charged carbon in a peptide bond in the antigen. So it forms a transient covalent complex, and a water molecule comes along, and the end result is that this peptide bond, this CO-NH bond, is hydrolyzed, with the result that we get fragments of the antigen at this step here, and we regenerate the catalytic antibody. So it can go on and on in multiple cycles of the reaction. In addition to the mechanism, the chemical mechanism of catalysis, studies from our group and from others have revealed that the non-covalent binding site is split from the nucleophilic site. So these are two amino acids shown in blue and green, and the green being the nucleophile that is capable of attacking peptide bonds. This resides in the antibody variable region, somewhat remote from the non-covalent binding site. This is important to point out because the catalytic activity itself is promiscuous. It can cleave a lot of different peptide bonds, but the non-covalent binding activity of the antibody for a particular target, so in this case amyloid beta peptide, is the reason why we get specific catalytic antibodies. And then again, work from our group and others has shown that that certain probes, electrophilic probes that are mimetics of the electrophilic carbon here. So this is a positively charged electrophilic carbon. If that is replaced with a phosphonate group, this group can bind irreversibly to the nucleophile in the antibody combining site. So this is important because the electrophile, the phosphonate electrophile, can be used to identify the antibodies and to pull out the good catalytic antibodies from a mixture of many different antibodies. Again, the notion of catalytic immunities, unexpectedly, the principles are somewhat contrary to what is known about reversibly binding antibodies. For instance, we and others have found that the mature antibodies, IgG class antibodies that are formed from these precursor antibodies of the IgM class by a process called class switching, the IgG antibodies are poorly catalytic. This is pretty much the exact opposite of what happens with reversibly binding antibodies, that IgGs develop high affinity binding for individual targets, whereas IgMs, because they are the first antibodies that are made by the B lymphocytes, they are comparatively unspecialized. In contrast, catalytic activity is greater in IgM than in IgG class antibodies. This is a reflection of the innate character of the catalytic activity. This is a function that is encoded by germline genes, and yes, it can improve adaptively, but we inherit the catalytic function. On the left is proof that B lymphocytes are that they can bind covalently to these phosphonate electrophilic probes for catalytic antibodies. So the green is staining for the B cell receptor containing antibody molecules that are complex to signal transduction, other signal transduction molecules. The BCR, or the B cell receptor, is central to an immune response because it must recognize the target. In this case, we are interested in amyloid beta peptide, and then that binding reaction results in cell division. The cell then proliferates and differentiates into what is called a plasma cell, capable of secreting the antibodies into blood. So the catalytic function is best found in IgM and to some extent in IgA, and it's difficult, the IgGs, it's difficult to find IgGs with the catalytic activity. Now again, I've included this slide to show an example of a pathogenic catalytic autoantibody. Indeed, this is how catalytic autoantibodies were discovered against a necessary self-antigen called vasoactive intestinal peptide, and we identified an antibody that breaks the peptide at the lysine and lysine-tyrosine amino acids. The catalytic site by mutagenesis was identified as a triad of serine, histidine, and aspartate residues, with the serine being the nucleophile. The oxygen of the serine becomes nucleophilic because of hydrogen bonding with a histidine imidazole, and on the right is shown the arrangement of these 3 amino acids on the surface of the antibody. The antibody catalyzes, it can be reused again and again to break the antigen. Because the antigen is broken, this is a permanent action of the antibody on the target, as opposed to a reversibly binding antibody which releases the target upon dissociation. So this is the reason why catalytic antibodies generally display greater potency than their reversibly binding counterparts. So we and others now have found catalytic autoantibodies to amyloid beta peptide of the IgM variety, and I am showing in this slide atomic force micrographs of non-catalytic IgM treated with fibrils, and the little dots here are the oligomers of amyloid beta peptide, and in the presence of the catalytic IgM you do not see the fibrils and the oligomers, indicating that there is retardation of aggregation, and preformed aggregates are also broken by the catalytic antibodies. The toxicity of the amyloid peptide in tissue culture is blocked by treatment with the catalytic antibody, as shown on the right side. By mass spectroscopy, the bonds that were cleaved by the antibody with a lysine-leucine bond and the lysine-glycine bond in the middle of the peptide sequence, as shown at the bottom. Young humans have only low levels of both IgG and IgM antibodies. The antibodies are increased as a function of age. So as the amyloid beta peptide is overproduced in older individuals, so are the catalytic antibodies. In Alzheimer patients, shown in blue on the right side, the antibodies are increased compared to their age-matched, non-demented controls. We interpret this finding of increased protective catalytic antibodies in the patients as suggesting indeed an effort to protect against the disease, but this is too little too late, and obviously while the antibodies might retard the course of the disease, the patients in the end do get Alzheimer's disease. Now, how are these antibodies, how do they come about? The normal reversibly binding antibodies, all they need is a new shape, a neoepitope. But for a catalytic antibody, we need something more. We need electrophilic stimulation so that the cells can strengthen their nucleophilic reactivity, which in turn causes increased catalytic activity. So in the disease, as I mentioned earlier, in Alzheimer's disease, a lot goes wrong, not just the aggregation, but there is increased oxidation due to advanced glycation end products, an example of which is malondialdehyde and glyceraldehyde, and the lipid peroxidation end products, the example being hydroxynoninal. These compounds, they can react with amyloid beta peptide, forming these keto modified groups. So this is amyloid peptide shown on the right with keto groups, and these keto groups are electrophilic. Here is a hydrazide treated amyloid plaque in the brain. The green indicates the presence of the keto groups. So amyloid beta peptide indeed is derivatized with these keto groups, and these antigens containing the keto groups can serve as the stimulus for B cells to produce the catalytic antibodies adaptively. So in this electrophoresis shown on the left, in the left bottom panel, this band shows the covalent binding of electrophilic carbonylated amyloid beta peptide. This is the hydroxynonenal derivative found also in vivo, and it reacts covalently with our catalytic antibodies, suggesting that it might act as the stimulus for the synthesis of the antibodies. Similarly, human lymphocytes shown in green can react with electrophilic A beta peptide. In this case, we have a phosphonate for this model experiment that I am showing you on this slide. And the notion here is that the covalent binding of electrophilic autoantigen can induce an adaptive gain of catalytic activity due to the mutations that occur in the variable domains of the antibodies as the B cells differentiate, eventually resulting in secretion of antibodies that can catalyze the breakdown of amyloid peptide. So our hypothesis based on the amyloid beta peptide studies is that old humans mount a beneficial catalytic autoantibody response to the misfolded beta sheet epitopes. Again, these responses are not going to happen unless there is misfolding, and then unless there are electrophilic self neoepitopes. So the response, we think, is driven by the electrophilic self neoepitopes to which the immune system is tolerant, and overall it is reasonable to believe that these antibodies are beneficial. We think that the natural ability to produce beneficial catalytic antibodies can be supplemented if we can find antibodies, monoclonal antibodies with a sufficiently rapid catalytic turnover and specificity for degradation of the amyloid beta peptide. So towards that end, we've used a library of antibody fragments. These are single chain FV fragments. Our library also contains, in addition to the single chain FV fragments, single chain VL2 fragments and then single domain VL fragments. The difference between these types of molecules is that we have a VH domain shown in blue. This is the variable domain from the heavy chain, and then a VL domain shown in red. These aberrant molecules, these single chain VL2 and single chain VL, they have only the light chains, and we and others have found that the catalytic sites are often located in the light chain variable domains. And for comparison, on the right I am showing you the structure of a classical physiological IgG with the constant domains and then the variable domains in color. So we pulled out, using our electrophilic probe, amyloid beta peptide probe, we pulled out highly catalytic antibody fragments, and we found that the best fragments were invariably either the single domain VLs, so these are only the variable domains of the light chains, or heterodimers of two different VLs. These are highly catalytic. Their injection into a transgenic mouse model of Alzheimer disease, the injection of these fragments directly into the brain results in clearance of amyloid peptide from the brain. These photomicrographs on the right show the needle track, and around the needle, as you see, there are sparse plaques stained with the antibody to A beta, but in the control, the plaques can be seen around the needle track. So the amyloid peptide can clear. The amyloid peptide can be cleared by injection of the antibody into the brain. We are developing stable forms of these fragments shown on the right. These are the VL domains attached to the constant domain. The purpose is to increase the half-life of the engineered antibody in vivo. The Fc fragment in the antibody stabilizes the antibody essentially by recycling. It takes in these Fc containing constructs into the cells and then puts them back out into circulation, thus prolonging the half-life of the antibody, as opposed to simply taking up the antibody and digesting it into small fragments. So this Fc stabilized antibody fragment shows an impressive turnover being the number of amyloid beta peptide molecules that are broken by the catalyst per minute, and it has a reasonable Km, which is an indicator of the non-covalent binding affinity. And we do not see cleavage of irrelevant proteins by these molecules. So these are sufficiently specific, we think, for therapy purposes. So the idea then is to increase the potency of the antibodies by isolating catalytic antibodies. They can be injected in the blood, much as the example of reversibly binding antibodies that I showed previously. If they enter the brain, they would digest the amyloid beta into fragments. The catalytic antibodies do not form immune complexes. Therefore, they will not be taken up by the macrophages, and they will, because the immune complexes are not stable, the risk of immune complex deposition and microbleeds as a consequence of deposition in the blood vessels is decreased. Now, to what extent are the studies that I have shown you on amyloid beta peptide, to what extent might they be generalizable? As I said, there are 27 different proteins that are known to form amyloid type of aggregates that are toxic, and I'm going to show you two examples. One, tau, which is about 55 to 62 kD protein, and phosphorylated tau forms these tangles shown on the left bottom of the slide within the neurons. By random screening of human monoclonal IgMs that we isolated from blood, we have found catalytic antibodies that break tau. On the right side, these are electrophoresis gels of biotinylated tau run on the gel, either incubated with diluent or with a monoclonal IgM. And as you can see, the tau band is greatly reduced. It essentially disappears. The small fragments, many small fragments are generated. On the right side in the electrophoresis panel, I am showing you an overexposed gel in which these fragments at lower molecular weight range are visible. We have also looked at transthyretin, the protein that exists as in the physiological state. It is a soluble homotetramer in the blood. it transports thyroid hormones, that is its physiological function, but with aging, the transthyretin tends to aggregate, and it is the cause of senile systemic amyloidosis. We biotinylated the transthyretin, and we tested the soluble tetrameric transthyretin and the fibrillar form of transthyretin as a substrate for catalytic antibodies. We found several human monoclonal IgMs that essentially cause disappearance of the biotinylated transthyretin. The fragments are visible in the middle panel, in the second panel from the left. These are very small fragments, less than 1.5 kilodalton. There is the amount of antibodies increases, as in the case of amyloid beta peptide, the amount of antibodies increases with age. So individuals 70 years and older, they make more antibodies than younger humans. IgMs are the best catalysts. IgAs, a few IgAs with catalytic activity were also found, and IgGs are minimally catalytic. So soluble transthyretin was not cleaved by the same antibodies that cleaved the fibrillar form. So here we have an example of antibodies that are specialized to recognize the pathogenic fibrillar form of the transthyretin. So it's fair to say then that with these three examples, that production of catalytic antibodies with increasing age might act as a beneficial mechanism against formation of amyloid with increasing age. The synthesis of the autoantibodies appears to be adaptive because of the age dependence, and there is some data that the electrophilic amyloids are the stimulus for production of the antibodies. Now, these autoantibodies, we think, act naturally as an anti-aging mechanism, but we get amyloid disease nonetheless despite the antibodies. So the clearance of the amyloid with these monoclonal antibodies that we are working with, monoclonals and recombinants, is a potential therapeutic approach to removing the amyloid. In addition to injecting antibodies into the bloodstream for therapy, there is the possibility of gene immunotherapy, which is really what will have to be done. A relatively cost-effective method must be found in order to make this kind of therapy realistic. Currently, a lot of antibodies, monoclonal antibodies, are used for therapy of human diseases, including cancer and autoimmune diseases like multiple sclerosis. However, the therapies are very expensive, and in order to reduce costs, the catalytic function, it is desirable because the amount of antibody that is needed to clear the target is smaller, but also a way to deliver the antibody genetically. It would be desirable where there is a consistent production of these antibodies implanted as vectors, as DNA vectors into, as an example, into the muscle. And indeed, some beginning evidence that, some initial evidence for the feasibility of gene therapy against Alzheimer's disease has been presented by our collaborator Ken Fukuchi, in which he found that antibodies that are expressed by way of gene therapy can clear amyloid peptide from the brain. In any event, as I said earlier, there is no cure at present for the amyloids. There is no therapy, no effective therapy. Catalysis by antibodies is a comparatively new field. The difficulty of the target, the amyloids, we feel justifies further exploration of catalytic antibodies for therapy, and as I explained, the preliminary data are positive. So with that, I want to thank you for joining me, and maybe I'll come back in a few years and explain further our progress on the antibodies. Thank you.